Swayam Prabha. Digital India. Educated India.
Dear students, today's class is something new for you. In this class, not only I going to teach you the basic sensing technology, their applications, at the same time I want to train you for the future research, for your future career, for so that you will be independent. So this course curricula is not for only the learning, at the same time you will be trained for your future career also. That's why this is very equally important, this course for your curricula. See, in this lecture I will cover the research proposal writing and the ethical clearance. What is this two? See, research proposal writing and ethical clearance, and these two topic I will cover because maybe, see, I already gave you so many things, right, topics. In this area you can consider, you can focus. Now, if you think about like writing a research proposal, and see, if you have to convince to some like other funding agency, or maybe if you want to go for like for research purpose, you have to say to your supervisors, means some your concept you have to write in the proper way. So you should have the proper information how to write your the whole concept. Suppose you want to develop a biosensor for, like last class I told, like soil health monitoring, right? So you have a very good concept. I can detect, I can develop some nanoparticles, it can monitor the health. Now you have to write some systematic way, scientific way. So that's why, as we are very close to end of this course, let's try to understand how you can write your concept and you can show this one to the funding agency or to your supervisor, so that you can start your internship or the small project, right? So that's why you should know this writing of the research proposal and the ethical clearance. What is this ethical clearance? See, especially the human samples, generally we are using human samples. What kind of human samples? Maybe blood, urine, or maybe saliva. So this kind of the samples we are using for the diagnosis of the disease. But this kind of samples, when you want to handle, because you have to drop this sample on a chip, and your chip can detect all the analyte. Your multiplex, maybe you developed a multiplex sensor, right? So that sensor can detect all the analytes at the same time. So it means your sample contains many contaminant. What's the contaminant? Suppose it can be HIV, malaria, or maybe dengue. So it means some infectious disease also can be there. You may want to detect malaria, but at the same time it can be HIV infected, right? So that you should properly follow your detection scheme and your safety protocol, so that if somehow your sample is contaminated, it should not spread to like we are healthy persons, we are like, you are the scientist, you have to detect all the like you want all the analyte in your laboratory. So you should not be affected, your colleague, that they should not be affected, right? So how you will handle those samples? So you should have a very good biosafety environment, right? So ethical clearance means once you have all the safety protocol, you know the safety protocol, you can do all the safety things before the sample measurement. If you have all these things in your laboratory, then you can go for this detections, right? So ethical committee, they will check that you should have those things or not, you are totally aware of these things or not, which kind of samples you are going to use. It is that also this committee, ethical committee, they will check. So you should have this kind of a very good information. I mean, ethical clearance is really important keywords for this course. That's why many times I told you, no, the many samples, it can be non-invasive samples or invasive samples, but they can directly or indirectly can spread the disease, no. That's why you should have the proper information. Okay, let's come first our first topic today, how to write a research proposal. So first thing you have to consider a topic. That's why today I just mentioned, let's think first which area you want to focus. So title, first you have to think about the title of your project, on which area you want to focus. So this one I will always recommend, like it should be innovative, it should be very much innovative. Then only you can go for some new innovations. I mean, say many things already have done, but try to improve those existing technology. What is the gap in this technology, and try to develop something new that can be useful for the mankind, for the society, or something rapid, something user friendly that can be useful for point of care testing, or that can be useful in the very extreme conditions, like in the high temperature, the high humidity, like on field testing, that you have to think. Why I am saying, as I told you, like enzymatic biosensor, right, that you know, but enzymes and different biomolecules, they can be denatured in the high temperature. So non-enzymatic, so you may think some very innovative project in the non-enzymatic procedures, because if you develop some nanoparticles, and if those particles are very much specific for the detections of any analyte, then these projects will be more innovative, something like this, and it can be used in a very extreme condition. So first think about the area, the topic where you want to focus. So let's show you how you can write the whole thing, your whole proposal. So let's follow this step by step, all the rules, then definitely you can make a good proposal. So first, let's say first thing will be your title of your research. This is really important. First you have to focus. Second is the objective. So your title ready. Suppose your title, suppose as I told you, like soil health monitoring. Now what will be the objectives? What is the meaning of the objectives? Objective means how you want to start. So the objectives, they may have different work packages. Work packages. What is this work packages? Means these work packages actually will guide you step by step how we can achieve, how we can complete the whole project. So there may be the many work packages, work package 1, 2, maybe around work package 10 you can make. So each work packages should have the all the information, step by step, all the information how you will complete your project. Let's give one example. Suppose you want to develop some aptamer based biosensors. So before the aptamer based, suppose you want to develop some aptamer. What is this aptamer? This is the receptor that specifically can detect the analyte, right? Suppose it can detect some analyte in the soil that present. You have to consider which analyte. That analyte specifically can bind with some aptamer. So some very specific sequence you have to develop. So your first job is the development of very specific aptamer. So this is one work package for your, and this will be on good objectives of your project. First objectives will be the development of this aptamer. So it can be in silico aptamer. So, and you can see I mentioned the deliverable. D1 means deliverable. So there may be lots, in a single work package there is lots different deliverables. Maybe you have to first validate your sequence, then you can apply those aptamer on the sensor chip. So all the deliverables you can mention 1, 2, 3, 4. Each work packages, it should have the sub points, they are deliverables. So first you develop the aptamer, then you can go for the validations. I mean, really this aptamer is ready for detections or not. Okay, so you understand what is the work packages. Then work package 2, you can come after the aptamer development, you just add this aptamer on a sensor device, right? So you can add this is the another work package in your proposal. So there is one deliverable can be in this work package, one deliverables can be your immobilizations of the aptamer on a like conducting polymer or maybe some graphene oxide modified sensor, right? You know, a graphene oxide modified sensor or maybe conductive polymer. So those can help to bind your aptamer on the surface. So this is one deliverable. So like conductive polymer fabrication on the sensing surface. And then you can immobilize the aptamer, and then after that you can go for the detection of the specific analyte. So this is one another work package. So these all the objectives you have to point out in the first you can summarize these objectives, then you can go to the full research plan. Very systematically you have to write. So first you have to think about the main topic of your work, then think about the objectives, means what is your goal, what is the aim of your project, just you have to write down. Then you can go directly to the research plan. So in the research plan it will contain, like as I said, different work packages. Work package 1, and it may contain different deliverables. Each work package may contain different deliverables. Okay, so before, like after the objectives or research plan, you have to think like 2 steps. This is A and B. This is very important. You have to write during the writing of your project. One is the state of the art and another is the review status. What is the state of the art? Means you are thinking some area to write or to start some work. So what is the research gap? Maybe there is many number of people all over the world, they are also working maybe in the similar area, but they are not focusing exactly what you are thinking. Not exactly, but something else also they are working. So there, it means they already published or they already have done some work, and you are going to start in some another area that is much better than their work. So it means there is some research gap. So you have to write those gaps. Then only people can understand the novelty of your research. Okay, this is very important. The state of the art and review status. So you have to review more papers that already published or already have done by other group, and let's find out the gap. So novelty. Novelty, this one you have to clearly mention by writing these 2 things. One is state of the art, what is already currently available, and what need to be done. So you can check like review status like national status, international status. Sometimes the problem may be very much localized problem. Maybe in this area only it is really important, and maybe not maybe equally important for all over the world. So you should have to think like which area this problem actually occur is really important. Nationally, you have to think also like whatever problem we are thinking in our country, how much important this one. Okay, so what about the national status? Is in all over India any other groups also working in this area or not? That also you have to check, and you can approach to your supervisor, or maybe you can, even if you want to start your own startup that I told you, you may have one R&D section there also. You can think in this area what is the gap. And international status means if you want to develop these things not only for your country, not only for our own nations, not for our India, our own country, maybe it can be equally useful for the outside other country also. So that also you have to think and how much research they have done on this area. Maybe they may already developed some. See, one area you maybe just thought that let's work on a glucose monitoring chip. I taught you many way. So some international group they already developed, but because of the patent issue, copyright, if you want to bring this technology to India, it will be huge, expensive. But if like made in India type, like indigenous, if you can develop something at cheaper cost, then it will be very much useful internationally. That's why it is not like this. They already develop. We cannot do something else. Yes, you can do something else based on the price. If you buy their technology, they want to bring everything to here. It is huge expensive. So you can think about how to cut down the price, right? That also kind of the good innovations. That's why you have to think about like different national, international status of the research. See, as we are very close to end of this our course, now that's why you should have this very basic knowledge of the plan that you want to start. Okay. Now all the work packages that I taught you, there may be another few work packages, but all the work packages should be very much clear. I mean how you are going to how to complete your whole project, and all the deliverables you have to mention very clearly. Suppose see the electrochemical performance analyzers, after the fabrications aptamer for the detection of the target analyte, maybe you are going to perform like their performance of the device, that step by step you can mention. It should be clear to your writing. Then testing of the real samples, then those deliverables you have to mention in this paragraph. Okay. Then finally you can go for the disseminations. You can you can publish the work, you can tell these ones to like the industry, you can approach and you can try to commercialize these innovations. That can be done in the end of this project. So one work packages can be this disseminations of the project. So this is just a format. It is not like the standard you have to follow this one. You can think some innovative way. It is totally up to your writing skill this way, but it should be very well organized. Properly you can think first and then start your new work. Clear? Now the Gantt chart. This is very see, I have just mentioned the example. I mean, if you want to start a research project, just make a chart like this. See, you can see here I mentioned like different work packages. First this is a column, one work packages, then all the time schedule. That you can schedule your time, then only you can successfully and very you on time to so that you can complete your project, right? So first 12 months you will do this kind of like first suppose you are developing a redox cycling method that I taught you. So those redox cycling method and like chemical amplification, signal to background ratio, everything can be done within 1 year or within maybe 6 months. That you just make a plan, then start your work. So it will definitely help you to predict some uncertainty. Suppose in this plan, suppose you are developing some chemical amplification technology, it may not work properly, right? So based on your plan, maybe you should have some other work packages. So if this plan is not work, you may think some other work package, some other plan. Accordingly, you can make this table. So you should have some backup plan also. If this work package not work, how you will move, right? So anyhow, you have to complete this work successfully. You have to complete. For that, you should have some backup plan. Those also you can just make a list, and all the deliverable you just make a list. Just see this plan and let's start. And M means the milestone. Means after this, suppose in this case, see, we are developing a redox cycling amplifications that will help to get the higher signal to background ratio. So the milestone will be you successfully completing the amplification strategy for your biosensor development. That is your milestone. So something like this way, one step you will achieve. One milestone you will complete successfully. Then go to the second work package and achieve the second milestone. Okay, so like this way you can complete the whole Gantt chart. See, then work package 3, work package 4, work package 5, all the work packages you have to make one milestone. Step by step you please complete it, and then you become a successful for this project. And as I mentioned that finally you should have a you can validate your sensor chip by using some real sample, and then you can disseminate your result to the public, so that your innovation not only it will be inside your lab, it should come outside your lab. So it should you can develop some device, so common persons, the layman, they should also use, so your society should be benefited. So this is highly recommended, because this course we design such a way so that this concept you can bring to the point of care testing device development. So we have to focus how to bring your concept finally on a chip, so that your innovation not only inside the lab, it will come to the market. So such a way you can arrange. You may need some collaborator, because this kind of innovations is highly collaborative, right? So then you have to make some your work packages, and maybe you have to go for the regular meeting with your collaborator, because it is very much interdisciplinary. Maybe you are biologist, you may know all the steps of the biological molecules, but you don't know how to integrate this sensor with some electronic instrument. So you have to collaborate with some electronic people. So you should have a regular meeting with them and collect the information accordingly. You have to make your whole work packages accordingly. Maybe you have to make one. So within this time, after 2 months or after 4 months, we'll meet regularly and we'll get the output and we'll achieve the milestone. Such a way you have to make. Then you can successfully complete your project. Okay, so that's the thinking, that's the thought actually for today's this proposal writing. Now next topic I'm going to teach you today, that is the ethical. As I told you, after writing the project, you have to think that kind of sample you are using for your sensor development. So if you really going to use some hazardous sample, some infectious sample, like some blood sample, like urine sample, saliva sample, they may contain some different kind of infectious marker. So that's why you should have the proper information. So ethical meeting, that's why you have to go through some ethical meeting. So who are the expert, they will critically evaluate your project, and then they will give you the permissions. Let's go for this development. Without their certificate or permissions, you cannot complete the whole real sample testing. So that's why your project should have all the topics. One is the sensor preparations, then calibrations of the sensor, then compare with some unknown samples and with the real samples. This unknown sample or the real sample case, you need the ethical clearance. So you please think here in this case, like the real samples that I just mentioned, it can be blood samples, right? If it infectious kind of disease present, so do you have the proper facility or not? Also to the ethical committee, you have to inform that how means number of the samples, how many samples you are actually going to collect, from where you are going to collect, who will provide these samples to you, and you have to make a consent form for those volunteers. Maybe some volunteers, they are giving the samples to you, so they have to sign in this consent form that's yes, they are giving the sample for the research purpose, something like this you have to make. So this procedure you have to complete. So that's why whenever you are thinking some device development with some this kind of hazardous real sample or some infection, it can spread some disease because of the sample handling. For that case, you have to properly follow all the rules and regulations, that is the ethical committee rule and regulations. Okay, this is very, very important. Otherwise you cannot complete basically your whole project. So another things, like samples can be collected from like different kind of patients, from hospitals. So definitely you have to collaborate with some, that's why, with some doctors. Why? Because your doctors know how to collect the samples. You are the scientist, you are developing the device, but you don't know how to collect the samples. Definitely you will not collect, maybe if you are not doctors. Definitely if you are not doctor, you are the very basic scientist, you will not collect the samples by yourself. So some people like doctors, they should involve with your project. So they will help you to collect the samples, and they will guide you how to handle the samples. Then let's add these samples on the device and check it. Okay, so this is the things you should have the informations, and then you are ready for the device development. Okay, so today that's why I taught, this is a very important part, the ethical clearance. So what are the conclusions for today's topic? So today I taught you first the write a research proposal. So writing research proposal is really important. Here you have to put your innovations, your idea, but just always think the novelty, right? Lots of research going on, but really this research is really important for the society. Let's think this, and think the gap. What gap? That is the research gap. Research gap. So you have to think research gap, national and international, right? So this way you have to think, like what is the research gap, then try to develop this your concept and start. So why, before the starting of your research, so you should have the ethical concept. What's the ethical concept? I mean, kind of sample. The sample you are going to use, is really there infectious? How to use the sample? For that, definitely you should have a collaboration with some medical professional, a medical professional or medical doctor. So they can basically help you to collect the samples, handling the sample. They can basically help you. So for that, you have to collect the clearance. So ethical concept, you have to collect clearance. Then you are ready to for the device development. Okay, so that's why these two things that I taught you is really important before closing this coursework. So let's prepare, let's think, and be ready for your own development. You become a independent researcher, and you can start, like I already mentioned before at the very beginning of this coursework. It will be the independent researcher. It is very much innovative for your own thinking. Like this way, you can start your thinking from today itself. Okay, that's all for today. Thank you very much.